All right, so I think we have a whole bunch of folks on and probably more joining. It's officially our start time. So thanks everybody for joining. My name is Dave Cooley. I'm the historian for the Cooley Family Association of America. And the organization started in 1936, and many of you are members already. And if you're not, check out our website. You can Google Cooley Family Association of America, and you'll find us pretty quickly, and look forward to interacting with you. We have been working with Michael for a whole bunch of years here as he's been working on the Y-DNA research, and he's here to give us a little education on the DNA, which can get pretty scientific, but we'll try to keep it high level today and talk about the differences in types of DNA testing and the Y surname project in particular and what it means to this project in particular. And that's, kind of researching the Cooley line. So the Cooley Family Association was founded by Mortimer Cooley in 1936. He was the Dean of Engineering at the University of Michigan, and his goal was to gather all the genealogical data of all the Cooleys in the US at that time, of course. But hopefully today we can learn some things about the DNA, and if you're interested, Michael can help us with any next steps. And if it's Cooley DNA related research that specifically might help us break through a roadblock, there is a small amount of funding that might be able to help support anybody that that might need testing, if it helps clear the path, if you will, for some results that'll be meaningful. So that's it in a nutshell. And with that, I'm going to turn it over to hopefully everybody grabs their coffee or their cocktail, and I will turn it over to our DNA guru, Michael Cooley.
Let's start. I first want to say that I'll be throwing out a lot of stuff out there, and I don't expect everybody to get everything. But that's okay. That's okay. I just want you to leave with a general impression as to how this works and how well it works. The Y chromosome is really quite simple to understand, but it's different from the others. 100% of it is inherited from father to sons and on again until the lineage becomes extinct. It's always been that way and will continue so. You might visualize it as a river that runs through any number of countries collecting debris on its way, mutations in this case. In fact, we might consider that we're here merely as hosts for the Y, that we carry it forward into the future, shedding bodies generation by generation, the Y chromosome, an entity all unto itself. Now, I don't mean to scare you with that, just making a broad analogy there. It's simple. But like fractals and algorithms that generate endless iterations, there's the occasional random modification that alters them slightly each generation, step by step. We see this in nature, rivers, coastlines, trees, clouds, and in humans. Similar but different. In time, the complexity grows and needs interpretation. But that's my job. To appreciate the Y, you need only to think in terms of rivers passing through multiple territories and altering the landscape. The Cooley DNA project was founded by Greg Parker at FTDNA in 2005. Only 5 years earlier, the company introduced affordable DNA testing to the world. I tested in 2006 and immediately saw its potential for breaking through brick walls. What genealogists have learned in the meantime is nothing short of astounding. There are limits, of course, with standard genealogical research. If no records are found, most of us are simply stuck. Others take speculation to an unreasonable level and offer that as fact, a problem that has become epidemic since the onset of the internet. And genealogists are necessarily focused on surnames, mere social conventions. And there's a difference between biology, of course, and and culture. A lineage's surname history, especially the changes in surnames, is often not known. For example, my Johnsons were once O'Neills. Why DNA told me that. I'd have had no clue. There's no record. DNA, particularly that of the Y chromosome, has all but eliminated those concerns. Something new is learned or confirmed with even the lowest level testing. In fact, the Y is an archive of genetic information that stretches back hundreds of thousands of years. We need only to understand how to read that repository. When doing so, we find that genetic identities are often better than surnames for tracking a lineage. Y DNA is a permanent record, more reliable sometimes than the court and government records genealogists regularly scour through. This record is found in every cell of every man's body. It can be excavated and interpreted much like an archaeologist does every day with bones and other ancient artifacts. This is a subject I love talking about and writing about. I jumped to the opportunity when offered it by the CFAA, the sponsor, as you know, of this presentation. Unfortunately, this will be much briefer than I would like it to be. Nevertheless, here we go. So just a summary of the Cooley groups in the Cooley DNA project. We're arranged by, an identification. CF merely means Cooley family, and they're numbered. Some numbers are missing because they got, pushed into another group. Some just, disappeared, no users, etc. They mean nothing above that. Please, everyone, mute. We're here for a genetic genealogy presentation and nothing else. Okay? All right. To continue, CFO2, the Tring Coolies, the Benjamin, what I call the Benjamin tribe, descendants of Benjamin Cooley, the Goshen Coolies. Goshen is a town in New York. Samuel Cooley of Connecticut, and Nehemiah and Joseph Cooley, who still live in the region of Tring, England. We'll talk about that in more detail. CFO2, the Pennsylvania cluster, the most recent common ancestor isn't known at this point. More about this, more about everybody here in a moment. The Spartan Coolies, brothers John and Edward of Halifax County, North Carolina. John's family went to Spartanburg. The CFO9 North Carolina cluster. These are names that I give to them. They don't have any significance beyond what I choose to give them. The North Carolina, the immediate origins were in Virginia. The MRCA is suspected. Irish immigrants, William and John. William went to Maryland, Pennsylvania, John to New Jersey. Were they brothers? Don't know. CF13, descendants of Joseph Cooley of Kentucky. He died in Howard County, I forgot, 1826 or 7. The descendants of Joseph Cooley of Kentucky. CF15. There's only one tester with a match, but I know nothing about that lineage. Not much work has been done. And Philander Cooley of New York, he's closely connected to a family named Bixby. Cooley could be descended from Bixby or the other way around. We just don't know yet. The genetic description, of course, that's what I'm going to talk about. Anyone positive for SK411, if you have that marker in your Y chromosome, You're of the North Carolina cluster. A few terms I'm going to talk about: SNP mutations, haplogroups and subclades, triangulation, despite the fact that it's misspelled there. And I've already talked about the most recent common ancestor, just commonly called MRCA. Okay, getting to the nitty gritty now. In every single cell, trillions of our cells in our bodies, there are 3 billion base pairs, 2 copies, one from the mother, one from the father, and they're all comprised of 4 genetic letters. They are A, C, T, and G. And for those who are chemists, that's how they're organized chemically. In this study, and in virtually every study, in fact, in most of the things that we do in life, we use comparative analysis. We compare one set of DNA to another set. Excuse me, I'm a little dry tonight. And we do this all the time. If you buy a car, you're comparing, its advantages, its disadvantages, but most, how are they similar and how are they dissimilar? Now, these 2 boxes look pretty similar. If you go along here in the second box, the first box, GAT, CAC, AGG, et cetera. But there's a difference. And it's this difference that we look for. Here it's moved from a T, it's mutated from a T to an A. That's called a SNP mutation. Just a single base letter has changed to another letter. And this happens at the inception of a new birth. One of the things I need to point out, of course, and you'll see more of this, the Y DNA is only inherited by men. So unfortunately, women can't take a Y DNA test, but your brothers, fathers, husbands, cousins, sixth cousins can do it. The cell, again, trillions of cells. We've got 2 basic kinds of DNA. The chromosomes are in the nucleus. The mitochondria are outside in the main body of the cell. They're extremely important. They provide the power for the cell. And what's really significant about that is each has a loop of DNA. And there are hundreds, depending on the cell. Fat cells don't need much mitochondria. Muscle cells need a lot of mitochondria. And if the mitochondria die, unfortunately, we will soon follow. But this loop of DNA has fewer than 17,000 bases, as opposed to the 3.2 billion bases in our chromosomes. So the 23 pairs of chromosomes that we find in the nucleus come in pairs: Pa, Ma, Pa, etc. We don't know which is which until we get in there and study it. The autosomes, chromosomes 1 through 22, we get it from both parents, and so there are 2 bases. Again, we have to figure out, not on an individual SNP by SNP basis. The algorithms do most of that for us, but just illustrate that both parents influences the autosomes and us. It's a vast majority of the DNA, but it comes in 2 pairs, or a pair from each parent. Mitochondrial DNA passes up the mother's line only. So the mother only influences the mitochondria. And so there's a single pair. And the same with the Y chromosome. All of this came from your father. In the case of a man, again, a woman will not have it. I'm not going to read all of this, just provide the bottom line here. It's good for up to 5 to 7 generations. And by the way, I almost forgot, at the bottom of the screen is this slide presentation. Actually, I need to update. There's an older version online right now. So you can go back and, but the autosomes are good for only 5 to 7 generations. The reason is that each time it passes down, you have the pairs from mom and from dad, and you keep getting less and less and less and less. By the time we get to our 6th great grandparents, we're lucky to have this percentage of their DNA. It will disappear. We will have no evidence of it in some cases, in many cases. Again, mitochondria, straight up the mother line, Y chromosome, straight up the father line. So, in other words, in autosomal inheritance, here's 5 generations. We can get our autosomes from any of those ancestors. Here's just one example. It could go this way. We could have gotten a DNA chunk from here. Anything is possible. And it takes a while to sort that out. Again, the computer programming will do much of this for you, but a lot of it you're going to have to do yourself by comparing with the DNA of various cousins and relations. Mitochondrial DNA, straight up the mother line. Y DNA, straight up the father line. So triangulation, it's much the same way that we triangulate the stars. 2 telescopes focusing on the stars. In this case, it's purely mathematical. Get the distance between the 2 telescopes, determine the degree of the angle as it's pointing to the star, and then you can get an idea as to where the star is. Of course, quite a bit different, but the idea is the same. This is an ancestor, Elias Pettit, born in the 1720s. My dad and very distant cousins triangulate to him on chromosome 16, a chunk of chromosome 16. There are other chromosomes involved there. Y DNA triangulation is much the same thing. Here's a 6th cousin once removed to me. We have nearly 100% of the exact Y chromosome, and we both have this SNP, YP4491. Therefore, we know that we're cousins. Therefore, we know that we're not part of CF02 or CF07 or CF13. This is a distinctly described family, which happens to be CF01. Examples of mutations here, I've given the SNP name, the defining SNP name for each group, the position on the Y chromosome that mutated, and the mutation itself, from a G to a T, from an A to a T, and et cetera, et cetera. Here's an example. This is just, I made this up. I make up a lot of things if you have to know. But I made this up. This is somebody who had a G on his Y chromosome or could have a G on his Y chromosome at, name the position, it doesn't matter. He had four sons. One of them was born with a C because it passes down 100% generation after generation. Everybody that follows him will have that C. Here later on, an A mutated. It'll continue, and in this example, most people remained a G. Okay, so this is CFO2. This is my attempt to describe the importance of haplogroups, which are simply collections of SNPs that we haven't figured out the timeline for each one. Each of these SNPs, by the way, occur about once every 4 generations. And I mean about. We really don't know. They can happen quicker, much longer, hundreds of years before SNP mutation arrives into the lineage, before that river picks up that little piece of debris and deposits it into the bay, if you will, which is our bodies. But they get parsed out. We sort them out over time, depending on whether or not we get additional testers. These SNPs are established cataloged markers, and it goes back thousands and thousands of SNPs, tens of thousands, hundreds of thousands of years. So when this Benjamin tester tested the big Y, I don't know that I've mentioned big Y yet, which sequences up to 15 million of these. He was found to be of this haplogroup, to have these catalog SNPs and everything that came before it. However, all of these were unique to him. They've never been seen before, never been cataloged. That's tester 1. Tester 2 came along, and what do you know? He matched. So this moved up to a haplogroup. It went up into the tree because we now have 2, and then later we found many more matches. But he did not match to this one. This one dropped off, and that remains a unique SNP to the first tester. Then a Goshen Cooley came along. He matched everything but these 2 SNPs. So these 2 SNPs came up onto the tree, or down from the tree, I guess you would say, and describes Benjamin Cooley, the DNA of Benjamin Cooley, and all of his descendants have these SNPs. The Goshen tester had 3 SNPs that was unique to him. We now have several testers. They all have these SNPs. So this becomes part of the haplogroup tree. Here is what CFO2 looks like now. Here's Benjamin Cooley. There's those 2 SNPs I talked about, descended from this haplogroup. If someone has these SNPs, they're almost certainly a descendant of Benjamin's. I say almost certainly because it's possible that his father had those SNPs too, that he got them from his father, and therefore his brothers would have them. So we've got 3 testers here with the Benjamin SNPs, but we don't know their lineage. Conceivably, they're from a brother, but I suspect not. There's no evidence of the brother being in the American colonies, not yet anyway. But I suspect that we will eventually find the lineages. It will help us to find the lineages if more testers come along and test for these SNPs, so we can get additional haplogroups. And when new testers come along, they sometimes bring a lineage with them. We've got the Goshen Coolies. Abraham and Jabez, an extra F in there. I tried to fix that before we started, but I didn't get it done. They were brothers, probable brothers, but it looks to be a near certainty. They all show up in Goshen together and left at about the same time. And another brother, Thaddeus, who moved on with Abraham. Jabez went to Ohio. Abraham and Thaddeus went to Virginia. Nathan is a descendant of this Daniel, who is not of Connecticut. That was a very big misunderstanding for a very long time, but we don't know their origins except that they have the Tring snips, almost certainly from Tring. We've got Peleg Cooley here, two matches. They match on this snip. Peleg is a descendant of Samuel Cooley of North Carolina. Long suspected to have Benjamin to have been Benjamin's brother, but I don't think so. I'll explain that in a moment. And then here, these descendants are still living around near Tring. The second one, Joseph just tested. I was hoping he would have some of Nehemiah's unique snips, but he doesn't. So, we're going to have to think about that to see how they were connected. They were close enough in age to have been brothers, but we don't have the genetic information yet. So the Coolies of Tring by family and era. Again, if you want to study this or copy it, you can download any of these. Just go to this URL at the bottom. You might want to make a note of that. It's on every slide. Here's Benjamin, son of William. Here's what we think is the CFO2 Samuel, son of Thomas. This is interesting. Thomas had a son named Thomas. That Thomas had a son named Thomas. That Thomas produced a Thomas the fourth. Whoops, excuse me. And he produced Thomas V, 1733. It would be really, really nice to find a descendant of that man. CFO1, this is my clan. In order to search for the deeper roots, I started a new project based on the YP4248 haplogroup. It's about 1,000 years old, probably a little older, and it happens to be of Norse origin, and that makes us think of the Vikings. You probably noticed I am dry, so take some Altoids here. Here are the Coolies and Whitfields, who are an exact Y chromosomal match to the Coolies. They definitely share the common ancestor in the near term. But we found some Higdons born in the 1650s. These two testers shared this haplogroup. They're also of my YP4491, but the advantage for me in this is that haplogroup was established by 1650. So it may go back to the 1600s, which means there were no SNP mutations between 1600 and John Cooley in the 1730s. More distant to Hawley and Hackett, but here we go. I'm also connected to this big group. These names, simple, story, Rankin, and all these Cochranes all go back, diverge in Renfrewshire, Scotland. They intermarried. So most of these, we don't know their origins beyond the early 1600s or so, but they definitely go back, or almost certainly go back, several hundred more years to Renfrewshire. So there are two groups of Vikings in that project. My group appears to be, I mean, this isn't proven yet, but now we're looking at the history a history that I was led to by the DNA. The paper would never have gotten me this far. Here's the Isle of Man, a troop, if you will, an army, if you will, of Vikings went up the river Mersey with Liverpool immediately to the north, and you can barely see it, but the Wirral Peninsula. They're referred to as the Wirral Vikings. The peninsula has Viking place name, Old Norse place names. And all those names that I mentioned, Cooley, Whitfield, Hackett, etc., they seem to converge in this area. So I've got a strong hint as to where my John Cooley came from. The second group, with the Rankins, Cochrans, etc., also part, probably, of this kingdom of man and the Isles. Another group sailed up the River Clyde, which I think is about here on this map. And here we see it. They sailed up the River Clyde, had a fierce battle, which they won at Dumbarton. Here's Renfrew. This is all Renfrew Shire, the county of Renfrew. Glasgow is just right there. Dumbarton is just northwest of Renfrew. This puts things into context. We can now look historically at things, and we have places now that we look to, which I never had before DNA. The CFO1 now is buried, and this was 2015. It's grown considerably, just to show how it all just kind of branches up and up and up and up. You can, when we have haplogroups and SNPs, we can begin to stack them to develop a timeline. It takes a lot of work to figure out this timeline, and it's not set in concrete. There's a whole lot of new information that we need that can vary this timeline, and it has varied since I made this a few years ago. And the clusters. This is L448 SNP. You get locality cluster. So with these Y-DNA SNPs, we can learn something about location and about era. And if we can dovetail in the surnames, all the much better. Okay, so I'll go through the remaining groups in the project really pretty quickly. The CFO4, we think they're brothers, but it's not been proven. Robert and Francis, who isn't represented here, were in, probably came in from, excuse me, New York into Pennsylvania. Richard Cooley was in Maryland, and here's another Robert Cooley. They all share this haplogroup, and this Robert, um, shares this haplogroup. It makes them somewhat distinct. Here we get a comparison again, similarities, differences. They differ by these four markers with these folks. And then I pulled in some Davises for context to give some, hopefully to be able to do eventually what I did with the CFO1 Coolies, get a historical perspective on it. CFO7, by the way, I have no hint that whether everybody's there or not. CFO7, um, these folks are closely related to these Coolies. These were brothers mentioned in their father's will. John went to South Carolina. I'm not sure about Edward. And we've got, these lineages that are closely related. Um, but enough on that. Let's proceed. CFO9 got a couple of distinct haplogroups there. James Roach is again closely related, and you can get an idea of that by counting SNPs and assigning about 100 years between SNPs. So if we count 2, 4, 6, about 600 years back to this. But we have to deal with averages. If we count all of them, divide by the number of testers, we get a more accurate representation. CF10. Same thing. We have little information about CF10. John and William might have been brothers, but we just don't know. They could have been first cousins, but they were undoubtedly closely related genetic wise. Here we bring in some Irish and Scottish closely related families, and William does have a record. There's a record that proves he was born in Ireland. So we need to look to Ireland to find the roots to this family. CF13, this is a descendant of Joseph Cooley, who most of us thought was a son of my John Cooley. And here he's got some Scandinavian matches, but it goes way, way, way back. This may go back 1,000 years. Again, ripe for study. We just need more testers, folks. CF15, very little is known. The relationship with Jackson is pretty ancient. And we've got 2, 4, 6, 8, 10, 12, 14. That's about 700 years separation. Again, that's not accurate. It just gets us into the ballpark. And CF17, he tested with 45 novel SNPs. Could take us back 4,500 years. He needs a second tester. There are matches to Bixby's. But we can't say that the Coolies or the Coleys in this case came from the Bixby's or vice versa. More testing. Okay, so here's the whole Cooley DNA project. All the way back to 30,000 years ago. Haplogroup P became haplogroup R. Excuse me. And that broke up into major haplogroups R1b and R1a. R1b is dominant in Western Europe, R1a in Eastern Europe and the Scandinavian peninsula. It's not common at all in Britain. A small percentage of the population is R1a. So we can see how everybody broke off into the various families. CFO2 has an ancient relationship, about 1,000 years, to CFO9. CF10 and CF13, according to this, uh, they go back about 3,000 years, and et cetera, et cetera. CFO4, which if I didn't point out, I don't think I did, may have had Viking origins as well. It's of the R1a haplogroup. Of course, the Vikings brought in more than just R1a. There were R1bs, there were Is, the whole shebang, if you will. But when we see R1a in England, we have to think Vikings. Okay, so myth busting. Again, I bring you back to my family. This is what really got me hot on the subject. I had heard about this book written by Laura Hamill. She put together the manuscript in the 1930s. Mortimer Cooley appears to have ignored her work, uh, and rightly so. She decided that a whole bunch of non-Benjamin Coolies were related from this Dutch man. I didn't put his birth here, but obviously, maybe 1650 or so. But look at this. It's got CFO1, CFO4 is represented. These are not descended from the same man. CF10, 13, CFO2, myth. And here we have, to finish it up, ftDNA testing. You can get started for $119, and you can $450 do the big Y. There are discounts for upgrades. Single SNP testing at YSEC for $18. So if you suspect you're of the CFO2 haplogroup, a man, could be a woman's husband or whomever, can test for $18. And they do whole genome sequencing at $315. It's everything. Women can do this too. Okay, I think that, one more thing. Here's some links. Again, if you go to this at the bottom, you can see these links, and I think this concludes the presentation. Let me see if I can hand back over to David.
Sounds good. Yeah, just unshare your screen, I think, or maybe keep it up because you never know, people may want to refer to it. How about that? Just leave it alone.
Yeah, I will.
That's great. So a lot here, obviously, to cover in a short amount of time. And, especially when you consider thousands of years of Coolies and how they're related or closely related or still need to be determined. And, it's fun for those that really enjoy the science. There are some that really just look for the results. That's kind of where I am. Michael knows the details, does the triangulation, knows the language, and we're lucky to have him kind of keeping an eye out for us and marshaling this whole project forward. So it's still a relatively recent science when you think about it, and it is kind of exciting. It's just you need a lot of time and the right skill set to dive deep, but it's fun, and I think thinking about Mortimer Cooley in 1936, and where we've come from where he was. It's just amazing. So our little organization, is welcoming any Coolies we do a reunion every couple of years in various parts of the country with typically nice research libraries or some Cooley historical connection, and we like to help with that, and meet each other. And Michael and I, as I mentioned in our little teaser, he's in the haplogroup that he and I are not related or don't have a common ancestor going back almost 25,000 years. So even though we're both named Cooley and we have funny beards, we're not necessarily related at all, and so that's where this DNA can really sharpen the focus for people who are doing their research work. So with that, check us out. Check out his blog. He keeps it up to date. If you're on Facebook, look at that DNA Worldwide Project. Follow both the Cooley Family Association and his page as well. And he'll update as results come in from testers. He pops up his analysis on there, and it's very interesting to read, even for folks like myself who can't really understand necessarily all the science. But with that, why don't we open it up as best we can here? Now, this could get to be mayhem, so appreciate everybody just staying muted. And if you have a question, let's see how we do. I don't have any way to open and close people's lines in particular. I don't think, Patty, unless you have a message.
Hi. So I think that Larry Cooley has a message directly for Michael. So I asked him to unmute and ask the question. And because the screen is shared by you, Michael, I can't see if there's a hand raised in order to address our audience, because it's all on the side of my computer. So let's just see if there's mayhem. And otherwise, if there's a pause, then please unmute yourself and ask a question to Michael if you have it. So Larry Cooley, would you like to go ahead?
Okay, hello. Michael, my question was, out of the list that you provided right there of the DNA testing, if you had to pick one or two, which would you recommend?
Pick them for what reason?
Which company?
Well, we've already done it through ancestor.com, but it's been a few years, and I'm wondering if we needed to update that, do it again. Is there another facility we should be doing it through?
Okay, well, if you want to do Y-DNA testing, and I'm sorry I didn't highlight that, FTDNA is the best place to go. Ancestry DNA doesn't do the Y chromosome. It doesn't do mitochondrial DNA. So you have to go somewhere else for each of them. FTDNA is really the master, if you will, for the Y chromosome. There are other companies out there, but I chose to mention only ysec.net. The prices are competitive with FTDNA. They don't have the group DNA interfaces like FTDNA does, and they don't provide matching services, so you'd have to upload to another platform to see matches. But FTDNA, they got started in 2000. They kicked all this off. They have all of those research tools available.
Okay, thank you.
Sure, thank you.
Would you say, Michael, that the best way forward, if somebody has a question, is to ping you for advice on where to go individually, or you pretty much have a handle on just go for the group that's interested in the Cooley family Y-DNA? Do you have a de facto place that people should go?
Well,
your life easier and make the project quicker?
Yeah, they have everything we need there. However, if you go elsewhere, I've written some code that analyzes the YSEC files, and which are all basically they present them in the same format. Sometimes I need to tweak. I had to tweak for the YSEC files, but conceivably I can do that for any company. It's a BAM file, by the way, BAM.
All right, I'm looking through to see if any hands are raised. Anybody have any hands they want to raise? And I'll hopefully see you perhaps. Case is on.
Michael, I'm a member of
Kay.
the, hi, we've corresponded many times.
Yeah,
And I'm from CFO7, which never seems to get anywhere. But I noticed that, you know, there are several different kit numbers in that group. And
there's
if
several different what numbers?
kit numbers that, you know,
Oh,
if,
kit
yes.
numbers, yeah.
So if somebody has a kit number that you are interested in, is there any way to contact that person?
So, yeah, well, do you have a relative who Y-DNA tested?
Yes. Well, we have a common ancestor way up the tree, but
Okay, but
I don't have a close relative.
do you have do you have access to his kit? Are you the manager of the kit?
No.
Okay,
Mary Winburn is.
okay. She can go into the account and find all the matches, she or he, I didn't quite catch what you said, and find all the matches, and the email addresses are there with the matches. I think you just highlight the name and you'll see that come up.
Okay.
Okay. All right. Thank you, Kay. Nice to see you.
Nice to see you.
All right, I'm scrolling through here best I can. If anybody has a question, raise a hand. I see Sean, I think is how you pronounce your name. And you're muted. Now you're good.
Hi there.
Hi.
My Coolies are on my dad's side, and there's really only one male left of that part of the tree. Is there any way I can get that information without, like, from my own DNA?
No.
No, it has to
No.
come from that male.
I mean, autosomal, you know, if it's within the first few generations, you might be able to find a match that will work. But to give you an idea, I've had 2 autosomal tests at FTDNA and 23andMe. I don't match to any Coolies. You know, so
I haven't
it
either.
doesn't help me in my Cooley research. So yeah, it has to be a brother, an uncle, you know, even the 5th cousin. I showed the example of my 6th cousin once removed. We have exactly the Y DNA.
It has to be a male, right?
But it has to be a male.
Male with a Cooley surname in the mix, and for this particular project, obviously there's a lot of other things you can learn in DNA, but for this particular project, you need to bribe your relative in some fashion, so he'll test.
Yeah, wonderful.
Right.
Yeah, there are some pioneers up here in Canada that founded one of these, a really nice part of Canada, actually, Upper Canada, built the first school and so on. So there's some pretty cool people. And anyways, I want to find the links. It's Preserved is my guy, as far back as I know.
Yeah, we've heard that name.
Okay.
Yeah, he could be a CFO too. He could be of the Tring Coolies. Some of them did go up to Canada. I'm not an expert on that lineage at all, but that's something to look for. And as Dave said, that's a familiar name.
There were several of them, like father, grandfather.
Oh,
Yeah.
Right,
born at sea, they were preserved in a storm. There you go.
Oh, interesting.
All righty, thank you so much.
Thank you.
All right, I'm scrolling again. Any hands? And if I don't find any hands, I'll
So I see in the chat Angie Cooley.
Angie.
Hi, thank you. Yes, so I have the Cooley is pretty dominant. I do have reason to believe on both sides. I do have a holograph of U5A. I don't know if that sounds familiar or any way to get any information from that. But
A
then
holograph? Did you say a holograph?
No, I meant I might have actually. I don't know what it's called.
Okay.
You know what I mean? It's the female, it's the mitochondria DNA. I did pronounce that wrong. I'm sorry.
Okay. Haplogroup, maybe?
Yes, that would be it. But it's
Okay.
U5A. And And I know my
Oh.
mother was a Cooley. And like I said, my father, I do believe there's a lot of stuff going on here that I'm finding out, but I've also found an association with me with the Cheddar Man. I don't know if the Coolies have any, if there's any history with the Cheddar Man.
Yeah, you're talking about mitochondrial DNA, which
Exactly.
goes up the woman, the female line, the mother line. We're talking about the father line in this case. Surnames typically don't go down the mother line, although a mother can pass one on, of course.
Well, like I said, there's been a lot of issues my father also was a Cooley, and there's a lot of things I'm finding out. They were apparently cousins. They didn't know. Long story, just Cooley is very dominant. So I don't know if that tells you anything.
Well, until you get a better fix on it, and the mitochondrial DNA isn't going to help you with that.
Okay, so what do you suggest? What kind of?
Your father still alive? He may not be.
I'm sorry?
Your father is still alive or may not be?
Actually, he just passed.
Oh, I'm sorry to hear that. So if there's
no brothers, cousins,
son or an uncle, or brother, that had the Cooley name, same line as him, if we could get a swab on that would give you the Y DNA results.
Okay, makes sense. Thank you.
All right, okay, thank you.
Let's see, Patty, you see any?
It looks like Kay has her hand back up.
Sure.
Getting back to my previous question. So I have the line that a relative has done, but if there are other kits that there's one that you say, hey, that looks like that's going to also connect to me, how can you get to contact the owner of the other kit that might be a connection? How can you contact?
You mean FTDNA?
Yes,
well,
yes.
Yeah, well, again, the contact for your relative who is part of the project, CFO7, I think you said.
Yes.
Get hold of her. Do you have her email address?
Yes. Now I can communicate with her, but that's not my question. My question is, there's another on that list, that under CFO7, there's a bunch of different kits.
Right. Again, have her access the matches in her account.
Okay.
And those matches will have the email address.
Okay. That's what I need.
For those individuals, yeah.
Okay. Thank you.
Okay. All right.
And I know there's a lot of folks who are not using their video, so it's hard to see your hand raised. So maybe just open up the line and jump in with a question if you have one. See how that goes. And Michael, you did a good job. I think at one point I counted 50 people online listening.
Kind of what?
We had about 50 people online listening.
Oh,
I think
good.
that there's a person named Tony Cool. Are you wanting to speak?
Sure, I can ask a question.
Excellent. Well,
I'm actually a Belcher, but my mother was a Cool, and I'm asking a question on behalf of one of my male Cool cousins. Our ancestor was James Cool, and he was born in 1829 in Franklin County, Virginia. And I suspect he might be connected with James Cooley, who was also in Franklin County, Virginia in the early 1800s. And the Cooleys disappeared from Franklin, Virginia by about 1825 or so. But my ancestor, James Cooley, he also went by Cooley, Cool and Cooley, in all of his records, census records, marriage records, and so forth. But he was born in 1829, and he was married twice, and on one of his marriage records, he listed his mother as Catherine Cooley, and listed his father as W.T. Clower. So
Hmm.
I don't know if he had a stepfather or if Catherine Cooley was her married name or maiden name. I've not been able to find anything about Catherine Cooley. But my question about my cool cousin is he would like to perhaps do the family tree DNA test.
Uh-huh.
If a Y37 test would be sufficient to determine paternity for either Clower or Cooley.
Mhm. Well, paternity is kind of a separate question. Um, One of the points I made at the beginning is that you can actually kind of forget the surname. The genetics will give you a portion of the story. So if he does a Y37 test, he's going to match, well, probably match, unless it's very rare, uh, other individuals of a particular surname. If there are Cooleys among those individuals, then that's probably his line of paternity. It won't tell him who his father is, if that's part of the question. So, yeah, a Y37 will match you with a specific set of testers. Sometimes, in my case, it's just dominated with Cooley. In other cases, there are a whole bunch of other surnames in that mix, which increases the likelihood that there was a non-paternity event somewhere in there. So, it's $119. It's not terribly expensive. So I would have him do that and just see what happens. Um,
Yeah.
and you're going to map, at 12 markers, for example, I match with people in India. All right. So the more markers you test, the more specific it will come. And Y37 it's been determined generally to be a good starting place. It provides enough information, to get your footing on.
Okay. Thank you.
Okay, sure thing.
All right. And then we have a question from Jason S. Feifel. Would you like to ask your question?
It looks like he's got the same autosomal kind of question, right? In order to really confirm the Cooley surname path, that you need somebody with that Cooley surname, a male, to do the testing. I'm just looking at your question, Jason. I'm not sure if you're able to unmute and if I'm answering it for you or not.
I'd like to, I
Yeah.
don't know anybody with the Cooley surname in my life.
Really struggling to hear you. I don't know if you're away from your microphone or.
In my case, I don't know of anybody with the Cooley surname.
Right.
It's a female.
Yeah. Yeah. I mean, again, the autosomal will tell you some things, but in terms of the lineage going back to which of the Cooley family lines, of which there are, I guess now 17 or so, we wouldn't really be able to pinpoint that, I don't think.
On paper, I have a testament.
Sorry, challenging audio there.
Yeah, yeah. Yeah, autosomes, they're not really part of the Cooley DNA project. As I mentioned in a early slide, if you go back several generations, you've got all of these ancestors, and you don't know the pathway to that ancestor, mothers, fathers, fathers, mothers, etc. Uh, the Y chromosome is straight up that male line, so it'll get you there. You might not necessarily have the name of the ancestor, but you've got his genetic profile. you know what his cells look like. And by matching up a number of people, the idea with any of this is that you're eventually going to find someone who knows that lineage and can identify that person. That can't really be done with autosomes. It's far more complex. Autosomal DNA is far more complex than ancestry DNA informs us.
Any other questions? Give it another minute. Go ahead.
Laura Cooley. Go ahead, Laura. Ask your question, please.
Did you ask me? Yeah. I'm sorry, I missed the presentation until a few minutes ago. So I'm not sure what all was covered, but I have two questions. One is that I heard somewhere along the line or in one of the communiques, something about the fact that some of the DNA did not check out. I'm wondering that Dean Mortimer had, Dean Cooley had traced way back when. And is that true? Is that the lines, like I have the Cooley, I guess the whole family line that he had mapped out. I have that CD and all that information. I was just wondering if some of those were wrong. And if that is true, who? I believe my grandfather does go right back to Benjamin.
Right.
And the other question is England, last I heard, when I was checked in here, Joan Arnott was thought to be the mother of Benjamin Cooley. And I wonder if that had been totally confirmed by DNA or no.
Yeah, that has not been confirmed by DNA. I think that's all been relying on records, and even that, I don't know that. And we've commissioned a couple of research.
I can't hear anything.
All right, are you guys able to hear me, Michael?
I can hear you, David.
Okay,
Yes.
maybe you have a volume control. Turn that up, perhaps. But anyway,
My sound. I'm not hearing anything.
I don't know where your speaker is or whatever, but check. Can you hear me, Laura?
I didn't hear any of that.
Okay, can you hear me now? Okay, so there's no way to determine through DNA that Joan is the wife of Benjamin or mother of Benjamin, I guess is more like the question that you had. So we're still, and we've had efforts to lock that down. I think it's pretty well published out there, but it's still a little bit of speculation from my understanding. And in terms of the book that Mortimer wrote, and a lot of assumptions were made by a lot of people, not just Mortimer, but there, some of them have checked out and some haven't based on the DNA, right? At the time Mortimer wrote the book, as I said earlier, it was thought you either went back to Benjamin or you were in this unplaced category. And through Michael's efforts and others who have tested, we know there's at least 17 lines, and not all of them are Benjamin related. So you have to really, if you have your grandfather or a descendant of your grandfather who's alive with the Cooley surname and have them test, that will tell you pretty quickly, I think, whether they do go back to Benjamin based on the results that we've got several good data points for Benjamin to be able to know that for sure. Michael, you correct me if I'm wrong.
That's correct. And I'd like to remind everyone that we have the two defining SNPs for Benjamin, A12022 and A1024, A1022 and A1024. Those can be tested individually for $18 at ysec.net. So if you just want to confirm a lineage and you've got the right tester, put up $18 and you've got your answer.
That's YSEC?
YSEC, SEQ, short for sequence. ysec.net.
Okay.
Michael, there's a question from Kent Cooley asking if you mentioned Iowa, and if so, which CF group might that be?
Mean the state Iowa?
Yes.
Oh, I mean, there are any number of Coolies in Iowa. It's impossible to say.
Okay, so
Yeah.
many of the groups could be in Iowa.
Yeah, they
Thank
could
you.
all be in Iowa for all we know. They're probably all in, represented in California and Texas. Yeah, you need the genealogy.
Thank you.
And the DNA too would help you.
Yeah.
I think that we're going to conclude. Anybody else?
Hi, one more question. This is Brian Coley. Michael, hi.
Hi, Brian.
I'm part of CF09, one of the testers. Right. And you mentioned the method of checking or counting the number of unique SNPs in the two lines.
Yeah.
You get 400 years, that is before the Common Era?
Well, it depends on how far back, and how many SNPs you're counting.
Right. I've got another tester that I'm in contact with. He has 3 unique SNPs, and I have 1. So that does that put us around 1623, 400 years?
Not necessarily. You need to know the SNPs and the haplogroup. You take the target, where you're trying to reach back to, count all the SNPs below that, including the private SNPs, count the number of testers, and divide the total by the number of testers. And then you can multiply by whatever year you think is the average generational whatever. I use 100 because it's convenient.
Right.
And some people say that the SNP mutation rate on average, and we're talking averages, is more like 82 or 83 years. And you just never know. You don't know until you know, right? Until you've got the final data. Then if you want to satisfy your curiosity, you can count the SNPs and then figure out really what the rate was for that lineage. So it doesn't matter that much what number you use. You're just going to get an overall possibility ballpark figure.
Let me ask this.
Yeah.
Is Brian connected through our data sets already, or can he merge his stuff into your project? So maybe that would get the answers, or maybe.
I'm actually one of the testers in CF09.
Okay, cool.
Yeah.
The chart he showed earlier, it has my kit on it.
There you go. Good.
And I'm just comparing myself to the other kits in this group. Some I'm closer to than others.
Gotcha.
Thanks.
Yeah, CF09 has so much potential. There are a good number of testers. There's been some bad luck in that, the haplogroups aren't forming as well as CF02 with the Benjamins, the Goshens, and et cetera, et cetera. You never know what's going to happen, but CF09 has a lot of potential. We can just get the right testers.
All right. Thanks, Michael.
Nice seeing you, Brian.
Good to see you too. Good to get a voice with that face.
Yeah.
Well, thank you, everybody, and it's really been a pleasure to see a lot of faces and everybody with this same interest. So keep in touch with our organizations, both Mike through his Facebook page and his blog that he puts together. You can Google him and you'll find him pretty quickly. And same with Cooley Family Association of America. We have a lifetime membership program. It's not crazy money, and stay in touch with us. And you never know not only do you meet people through the DNA, but just through getting together and sharing stories. So thanks so much, everybody. Patty.